Hello, today we will discuss about the protein digestion. So let's begin to understand what is protein first. So this is the food, this is the chicken which that's contain protein. So the protein is made up of amino acid chain is known as polypeptide. Poly mean many and peptide means CN bond between the amino acid, and in this way the polypeptide will be formed. Here is the amino acid 1 which contain alkyl group 1 and alkyl group 2 in the second amino acid, and third that is basically is the N terminus and C terminus. The further on in this way the amino acid will be bind one by one with the C terminus or N terminus. This is the structure of the protein. So let's begin to understand. First is the digestive system, and then we will understand the digestion of protein. So the amazing thing is that essential amino acid and non-essential amino acid. Essential amino acid means our body will not synthesis from the dietary amino acid, while the non-essential amino acid mean our body can transamination to convert the non-essential amino acid to a non-essential amino acid from the dietary amino acid. So in this way our body can synthesis. So that is known as the non-essential amino acid. While the essential amino acid mean our body not able to synthesis. So this is the digestive system contain parotid gland, sublingual gland, and some submandibular gland. This is the salivary glands, and here is the stomach, pancreas, small intestine, rectum, large intestine, and cecum, as well as appendix and anus. So this is the whole digestive system. Liver also involved, but here I am not drawing the liver structure. So let's begin to understand from the entry of the food after the oral cavity. So in this way the esophagus is very important, contain a circular and longitudinal muscle for move into the stomach, through a gravity also play important role. So in this way this is the esophagus. After the go through cardiac sphincter are present, which that will be open to move the food into the stomach. After the entry of the stomach, so the stretching of the stomach wall will produce through enteroendocrine cell to produce gastrin, and gastrin released into the blood circulatory system. Remember, so this is the chief cell, parietal cell, and enteroendocrine cell. That is basically the gastric pit contain these cells, and these cells in the form of gastric pit. I am not drawing that gastric pit. So here is the pepsinogen. This pepsinogen when released from the chief cell and move into the stomach, when increase the pepsinogen, that will lead to also produce the parietal cell hydrochloric acid. Hydrochloric acid is responsible for the pepsinogen conversion into the pepsin active enzyme. So when active enzyme, so hydrochloric acid will lead to low pH in the to increase the pH, sorry, not decrease, low the pH for more acidic. So it means the amino terminal side peptides bond hydrolyze in this way through pepsin. But this is the partially digestion of the protein, not completely, but the efficiently digestion will be occurring in the duodenum. When the food will go into the duodenum. In this case, here is the gastrin release after this stretching will lead to increase the more positive feedback to the chief cell and parietal cell for increasing the pepsinogen and hydrochloric acid, for increasing the more activation of pepsin, and in this way the pepsin activation from the pepsinogen will lead to more digestion and more hydrolyzation of the amino acid. So let's begin to understand from duodenum here. So after this digestion will be occur in the stomach, will go into the duodenum. This is known as chyme. This chyme will go into the duodenum, and this duodenum contain brush border cells, enterocytes, and enteroendocrine cells. So in this way, here is the protein, which that is semi-digested protein. After this is basically is the acidic content low pH in this way, because acidity will be increased due to the hydrochloric acid. So remember, so after this, here you can see this is the pancreas is very important, play a role for the digestion of the protein further in the duodenum. So the pancreas will produce the trypsinogen, procarboxypeptidase A and B. In this way, you can see this is the trypsinogen, chymotrypsinogen and procarboxypeptidase, which that is the inactive. But the pancreas also produce the not pancreas, but enterocytes will produce enteropeptidase. This enteropeptidase is containing protein containing trypsinogen, chymotrypsinogen, and procarboxypeptidase, which that will be hydrolyzed, and in this way the modification to through our trypsin. This trypsin is also released from the pancreas, will be convert the chymotrypsinogen, and trypsinogen will into the chymotrypsin and carboxypeptidase A and B activation, and trypsinogen will convert into the uh, uh, trypsin from enteropeptidase. So the trypsin will important and play a role for the further activation of the chymotrypsinogen and procarboxypeptidase. So all these enzymes will release from the pancreas exocrinally into the lumen of the duodenum. So in this way, here is the complete the hydrolyzation. The hydrolysis the protein efficiently will lead to dipeptides and tripeptides and single amino acid. This free amino acid will be converted through a brush border enzyme dipeptidase and tripeptidase. This dipeptidase and tripeptidase will convert into the simple amino acid, and simple amino acid will be moved into sympore transport, and this sympore transport is known as secondary active transport, and in this way the proton will move in with the one amino acid. So the two amino acids is also moved into the enterocytes. So again the sodium will be moved out with the sympore transport with the proton out. So here the sodium will enter, so the one amino acid will be enter is known as sympore transport. So the hydrochloric acid will convert into the chlorine and proton. In this way the chlorine will increase, so the low pH will be decrease, not increase. So in this way the increase of the pH, it means the basic more. So the enteropeptidase is here, and in this way here is you can see dipeptidase enzyme further can be digested through inside of the enterocyte is the dipeptidase enzyme. So in this way here is the primary active transport required the ATP. In this way the sodium potassium exchange will lead to move inside the amino acid into the blood circulatory system. So this is the interstitium between the circulatory system, blood circulatory system and enterocyte. So this is the facilitate diffusion, and in this way the amino acid will be moved inside, will lead to increase the amino acid as well as glucose, but here we will not understand about the carbohydrate digestion. So also the enteroendocrine cell will produce the cholecystokinin and secretin. This cholecystokinin and secretin will produce, and in this way it will be targeted to the pancreas. After this targeting to the pancreas will lead to increase more and more enzyme and secretin and exocrine hormone enzyme. So also livers when contain the insulin binding with the receptor will lead to entry of the amino acid and glucose, and in this way you can understand this is also the sympore transport. So anyhow, this is through portal vein. So because the digested food when assimilate will move first into the liver through portal vein. So here you can see this is the pancreas, and this pancreas which that is basically cholecystokinin and secretin, which that will be bind with the pancreas will lead to increase the bicarbonate, trypsinogen and chymotrypsin chymotrypsinogen, procarboxypeptidase. This all enzyme which that will lead to digest the food which that is we had already discussed before previously in this video. So let's begin to understand the metabolism of amino acid into the liver and what is the role of the amino acid. So let's begin to understand first the metabolism of amino acid into the hepatocyte. So basically when amino acid will be moved inside through facilitate diffusion, and in this way the amino acid, for example the transamination will be occur, and in this way the alpha keto acid is the mediator molecule, and this is the basically the C skeleton which that it will be remain, but the other modification will lead to increase the glucose also. Let's begin to understand here. This is basically you can understand again. This when amino acid will be moved into the cell hepatocyte. So the hepatocyte is very important and play important for role for the beta oxidation, and there are a lot of things. So this is the glucagon receptor as well as amino transporter. So in this way when amino acid into the cell move into the cell, it will transamination will produce the alpha keto acid. This alpha keto acid which that will be C skeleton, and in this way it can be converted into the glucose, either glucose or go into the tricarboxylic acid cycle and to produce amine, and this amine group basically move into the urea cycle through here is the basically the fatty acid conversion from keto acid also and acetyl CoA, and in this way the lipolysis will be decreased because we are going to fat state, and this is anabolism, and anabolism means the synthesis of protein, not degradation or synthesis of the variety of the molecule. So here is the urea cycle which that will be decreased and urea will be decreased, and beta oxidation also decreased because it is the catabolism. Autophagy is also decreased and mitophagy also because it is also catabolism. But the anabolism will be increased, mean the amino acid in the cell which that will be synthesis of an containing compounds, hormone, GABA neurotransmitter, and on the other hand the glutamate, a neurotransmitter, as well as cytokines. Cytokines is very important for the inflammatory response and inflammatory mediators which that will produce from the cell. So that is why it is also come from the protein out of the synthesis from the transcription to translation into the cytoplasm. So on the other hand, these all things will lead to uric acid production when the water will require less to urine, and in this way the uric acid will produce more after this catabolism. But in this case here is a glucagon receptor. When glucagon will be burned, which will lead to this synthesis of the varieties of the molecule will be decreased because the